Did you attend this class? Here or anywhere? Okay. I will have another class in the afternoon with which group? The last one is the fuel group that I have a lecture.
I'll watch the recording.
Entirely up to you, sir. Right. Can you all hear me?
Yeah.
Sir, can you hear me? Sir, can you hear me?
واحد وراك يطلع السلام
This really takes two minutes. You see, like, there's a lot of work. Like, you come around.
لا شو يقدر يدخل من وراك شوف بعد الإيميل
All right, guys. Challenge. We'll finish around 5 minutes past 12 then. Okay, we're starting at 10 minutes past, so we'll finish in 55 minutes, inshallah. We should be settled. That's a young man. Did you guys have the chance to go over the Cell 1 lecture? Yes. Okay, good. Now, did you have the chance to go over the Cell 2 lecture as well? No. No, not yet? All right. Not too late, but make sure that you finish it as soon as possible. Now, these are the objectives that I shared with you in the last lecture, and the main focus of this lecture would be cytoskeleton and the nuclear core structure, nuclear membrane structure, and how the chromatin material appear like in light microscopy. So what is U, what is heterochromatin, and what are the main steps in the cell cycle. Okay. Now, what function you can assign to cytoplasmic cytoskeleton? What function do you think that the cytoskeleton of the cell performs? So, protection. So it forms a body of the cell not resilient, not weak enough to be damaged. So hence you are protecting it. Okay. Sir, at the back.
Maintains structure.
It maintains a structure, maintains the shape of the cell, maintain the structure of the cell. Okay, so maintain some kind of asymmetry of the cell. Okay, so the nucleus has to sit here, not keep moving. The nucleus has to stay there, and the mitochondria will be there, and the microvilli should be on the top. The cilia should be on this side, not the other side. So this is what the shape. Okay. Anything else that you can think of, sir?
Help with the movement. So,
Help
of movement in the cell.
help the cell itself to move, and also move the material within the cell. So it will help the cell to move. There are cells which are running in the blood. They might squeeze between the blood, between the cells of our capillaries. So they can squeeze out. So they can help them do that as well. Okay? They maintain the shape and stuff like that. Yes, sir.
Also interferes with mitosis. It's
Excellent. They also help the cell in cell division, and we'll see how. They also help in cell division. Sir. Sorry?
Rigidity.
Rigidity and the fluidity. So they are basically the points of anchors for the proteins. Remember we talked about two types of proteins related to the plasma membrane? Yes, no? Peripheral and
Transport, yeah.
transmembrane or integral proteins and peripheral protein. Now there will be certain proteins which, for instance, form the junction between the two neighboring cells. Okay, so they are, but inside they have the cytoskeleton which is supporting the junction. Okay, that makes sense. So we'll see when we learn about the epithelium and the junction between the cell and see that how the proteins are being reinforced from inside the cell. Reinforced from inside the cell. Okay, and this is basically a function which our skeleton, a vertebral skeleton performs as well. Help in structure, movement, polarity. There has to be the head and the trunk and the legs and the arms. You see that there is a structure. What are the three elements of cytoskeleton or cytoplasmic cytoskeleton? Three. Microtubule, microfilaments are actin filaments, and intermediate filaments. Intermediate filaments, the size is between microtubules and microfilaments. Let's look at this. Uh, let's look at this drawing. Now, different types of cytoskeletal elements are being shown here. Okay. Now, these are the microvilli sitting on the top of the cell. Look at what is sitting in the core of the microvilli. We have the actin. Look at your own finger. It has the bony skeleton inside. Think about it. There is no bone inside your finger. What's going to happen? Yeah, it will be hanging like, hanging like a deflated balloon. That's it, because there is no strength to it. Similarly, the undersurface of the cell has the intermediate filaments which are maintaining the shape of the cell. Okay, they are maintaining the shape of the cell. Does that make sense? So you see that. Now I want you to look at the blue ones, which are the microtubules. These are the microtubules. Can you see this vesicle here? Can you see the vesicle there? Yeah. Okay. And these are the two proteins which are related to the vesicle. And this vesicle is as if it is moving. Can you see the purple, dark, and the blue and the purple color vesicles as well? You see, as if they are moving on the microtubule itself. That makes sense? Yeah. What will be the function of the microtubules? They will help movement within the cell. So basically, we talked about the different substances that are produced by the cell. For instance, neurotransmitter, which is produced in the cell body of the neuron. But the exon is a very long process. The neurotransmitter needs to be released there. How would the vesicle then go all the way there? Microtubule will help us. Microtubules are like train tracks. Okay? They will guide, go this way. The vesicle will go that way and will be released there. That makes sense? Had there been no microtubule, the vesicle will not way to go. Sir. Cytoskeleton for the time being, not only active. Do not just extrapolate exactly the word to word meaning. Okay, this is just an analogy, not accurate translation of things. Just an analogy. Now look at this diagram, which is basically fluorescent micrograph. Different cytoskeletal proteins are being stained with immunohistochemical method, and then you see that's where the nucleus, and you see the network of microtubule and skeleton that you can see sitting all around. Function, we talked about shape, important role in organelle and cytoplasmic vesicle movement within the cell. Also, the movement of the cell itself. All of them. Do not just say one, but this at this point in time, everything's together. Okay? Now, microtubule. Let's look at the microtubule. If I ask you to imagine a rope and imagine a tube, what would be the main difference between the two? The tube will be hollow inside, and the rope would be a solid structure. Think about a rope which is also twisted. Okay? The threads of a rope are also twisted. Now, keep in mind, keep this imagination in mind. So, if you look at the microtubules, they are long, hollow, unbranched tubes composed of subunits of protein called tubulin. Now, if you look at the tubulin, which is alpha and beta tubulin heterodimer, look at this one, alpha and beta. These two are heterodimers. Another dimer, another dimer. So these are the dimers. Okay. Now these dimers are organized round the circumference, 13 of them, but they are arranged not at the same level. Not at the same level. They are arranged at, like staggered. Do you know what the meaning of the word staggered? For instance, can I borrow this? This one and this one, they are sitting at the same level. They are registered at the same levels. But what if I put this phone at this level? The next phone will then sit at this level. If they keep on moving, then we move, make it a circle like this. So you see that these two phones at the end will not be matching and aligned. So you see this dot here and this dot here. So they are staggered. Look at this beta subunit, which is not at the same level of beta only, but between beta and alpha. Look at this alpha, which is between alpha and beta. But this whole ring is that of beta. This whole ring is that of alpha. Because of this, we have this helical arrangement. We have this helical form around the circumference of the microtubule. Because the way that the dimers are polymerizing, we have two ends. One is the positive and the other one is the negative end of it. Positive and the negative end of it. Okay. Now, centriole and centrosome. Now, if you look at the microtubule, can you see a microtubule here? How many microtubules are sitting together? Three. Now, three microtubules together are triplet. Now, 9 triplets sit around together. That structure is called a centriole. That is structure is called a centriole. You understand that? Okay. So centriole are paired, short, rod-like cytoplasmic cylinders made up from microtubules. Okay, 9 microtubular structure. Now, centrosome is the collection of these centrioles, okay, where the microtubules are being organized. Okay, so centrioles will have two centrosomes along with certain protein around it to form what? To form the microtubule organizing center. So this microtubule organizing center is a region where most microtubules are formed and they are directed to different places. The microtubules which are sitting in the cilia, where are they produced? They are produced in the centrosome. That makes sense? So that microtubule is formed and then went there. Now it's in there, attached with another structure there. Not the whole centrosome will sit there underneath every cilia. That makes sense? Are you following what we're talking? Organized. Centrosome will form the microtubule and then they will go to their places. What will be the function? What will be the function of the microtubule? Yeah, we talked about it. Vesicular transport, secretory vesicle, endosome, lysosome. They are all the vesicles, aren't they? So they will be moving around. How would they move around? They don't hang in the air or fluid and then they can then swim around. No. They have to have certain proteins on the surface of those vesicles, which is attached onto the microtubule, and then they move around.
They said it's coming.
It's coming? Thank you, sir. Sir?
Just the centrosome. The centrioles contain the centrosome.
Sorry?
The centriole contain the centrosome.
Correct. Centriole is the region where the centrosomes are present, along with certain other organizing proteins. Sir, you have a question.
I'm a bit confused on what the centrosome is. Could you explain again?
Centrosome. What is this podium? A computer, a microphone, part of a desk there. Together is podium. Centrioles come together along with other organizing proteins with that centrosome. That's where the protein, the microtubules have been formed.
Okay. So a lot of centrioles is a centrosome.
Two.
Two centrioles is one centrosome.
There you go. Okay. That makes sense.
Yes.
Look at the network of these tubules, microtubules in a cell. It can help. It does help in elongation and movement of the cells. They help in movement of the cilia. They help in the movement of the. So they form the core of the cilia. They form the core of cilia. The core of the the villi is formed by another structure. They also help in movement of the chromosomes. Okay? They help in moving the chromosomes. As if the chromosomes crawl along or pulled along with the help of microtubule. If there is no microtubule, or if you block the formation of the microtubule, would there be any cell division? Sir? Why not? Exactly. They have to, the newly duplicated chromatin material have to move towards the center of the daughter cell, and this is helped by microtubule. If you block it, the DNA will be duplicated, but the cell will not divide. Sir. Spindle fibers are forming many of the microtubules together. Okay. Now they also maintain the shape, particularly asymmetry. Asymmetry means the cilia needs to be on the top. Okay, the cell has to be long. Cilia will go there. Cilia will come. This is what the shape. The cell has to be disc-like. Okay, not rounded. Maintained by. So, what is this? So, can you tell me what is the central here?
Me?
Yeah, I told you.
The center of the chromosome.
Central here is the point of the chromosome. Between the long erection of the chromosome. Okay, that's your. We'll come to that. We'll come to that when we talk about it. Okay? Chromatids are joined together at that point. Okay. Now, excellent.
Of cilia and that we would use it. So how do you know
Look at me. I'm a cell. These are the two cilia. The core, the center of it is microtubules. That will help me move like this. But there is a vesicle inside my tummy that has to move as well. Microtubule sitting there will help it move there as well. This is how the cilia and flagella move. This is how the vesicles move inside the cell. Okay. Now, intermediate, the microfilaments, which are actin filament, these are around 8 nanometer diameter, and they are composed of protein, which are globular protein, actin, and they are the most abundant ones. Okay, you must have learned about the different types of protein molecule, globular and linear shape of those. Okay, fibrin, okay, like a thread like globular and fibrillar protein. This is a globular protein, actin. Is that globular? Now, in presence of ATP, means in presence of energy, the actin monomers they polymerize to form the filament, the helical arrangement of the filament. You see that helical arrangement of the filaments? Look at these globular proteins. They connect together in presence of the energy, and they form the polymer of this globular protein called actin. Okay. Now this actin will also have a positive and a negative end. Now this is the negative end. How you see the negative end there? Well, that's where they are being kind of cleft. They are open. So as if you are twisting something, and there are two, and then if you take two ropes and keep twisting it like that, the one end which is open is the one which is the negative end of it. Okay. Now, what are the functions of these microtubules?
Oh yeah. So the actin, how many subunits is it made of? Yeah. Oh,
Many globular proteins come together, fibers, and the fibers are chemically aligned. Yeah. So, where is that? Yeah, the micro. Where is that? It makes noise, but then okay. Now let's look at the function of they are anchoring the transmembrane, the protein, the membrane protein. Remember we talked about membrane protein? There are some membrane proteins which will be integral, the others are peripheral. Okay. Now you will learn in epithelium, the two cells will have junctions together. Okay, so there are proteins which are coming together from two different. Inside of the junction, there will be these actin filaments which are supporting those. So anchoring and the movement of the membrane protein. Also the locomotion of the cell. You see that? So it's not only one. There are different things of the cytoskeleton which will help you. Formation of structural core of microvilli. Okay, the microvilli will have actin. Actin does not bend. Actin doesn't bend. So should the microvilli bend?
no.
That's why the microvilli stay stiff. If they stay stiff, these are the ones which will help increase the surface area of the cell, for instance, for absorption of nutrients. If you need to increase the surface area, your microvilli, core of the microvilli comes from actin. Intermediate filaments are in between the two tubules and actin filament. Rod like is starting from a monomer into a dimer, okay, and the two dimers come together in anti-parallel direction, okay, in anti-parallel direction to form a tetramer, okay. That's how we form the intermediate. Now they are also uh, responsible for stability of the cellular structure. Okay, it's not that if you pinch something, the cell will change its shape. Okay, cell will bounce back, it will retain the same shape again. That makes sense. Now they made up of the protein subunit of different cells in different type, different subunits in different cells. Okay, different protein subunit in different cells. If you know this cell type will have this kind of intermediate filament, then if any problem of that particular cell arise, if you see that protein, you know that this pathology is coming from origin of the cell from the skin. This tumor is coming from nervous tissue because we see this protein in the neurons. We see this protein in skin cells. So if you see a cancer which is spreading from one organ to the other, you take this sample of the cancer, you look for these proteins, and based on that you can tell, this is coming from there, not there. This is coming from source A, not B. Okay. There are many classes, but these are the 6 major classes based on the structure of the gene responsible for that particular tissue, and then protein composition itself, and the cellular distribution where they have been distributed. Now let's look at keratin. Class 1 and 2 keratin protein. Where the keratin protein is commonly found? They are in skin. Keratinocytes only. Okay? That makes sense? Now if you see some pathology, often of the skin. Okay? If you see that, it's stained with the keratin. You say this is coming from ectodermal in origin, it's coming from the skin. Then you see class 3 protein. There are 4 proteins: vimentin, which is widely distributed, intermediate filament in the body, and vimentin-like proteins like desmin, glial fibrillary acidic protein, GFAP, and peripherin. Type 2. New, sorry? They are mostly found in that in the body. They are mostly distributed in. Okay, they are most widely distributed. Means they are present in many. Now, if you look at this neurofilament and nestin, they are present in axons. Now, if you see a pathology having keratin, is it coming from neuron? If you see a pathology, a tumor having nestin or neurofilament protein, it's a neuronal region. You see that you can differentiate. Lamins, which are located within the nucleoplasm of almost all differentiated cells in the body. Now, if you see lemon, would you be able to tell that this pathology is coming from the skin or other? No, because it is present everywhere. That makes sense? We need to go for what is specific in a specific area. Make sense? Yes, no? Yeah, that's how the modern day pathology is helping differentiate. Nucleus, what it is a command and control center where the chromatin material is present along with the molecular machinery. So DNA and its replicating machinery which is present, that's called as nucleus.
What's the difference between keratin and cytokeratin?
Keratin is a protein. Cytokeratin is something which is cell after the therapy inside cell. Keratin. Okay.
Did you see last name?
Sorry.
Yes.
No. Yeah, the class 6 is written there. Class 6 proteins are present in the lens. They are the lens specific protein. Again, lens protein. Nucleus, conventionally, it's oval, it's sitting in the center. Having DNA, having the machinery for its replication, means certain enzyme protein are sitting there. Okay. Now let's look at this H&E preparation. That's where you see the nucleus. Okay. That's the cytoplasm. That's where the nucleus is. Okay. Now, nucleus is surrounded by a membrane. We call it the nuclear envelope. We don't call it the nuclear membrane. We call it a nuclear envelope. Means envelope will have 2 layers. So there are 2 membranes. So it's selective membrane barrier, okay, which is between the nucleus and cytoplasmic compartment, and it has 2 membranes separated by perinuclear space. Look at this material here, chromatin and everything, surrounded by a double layer here. You see the membrane here and membrane here? Can you see the 2 membranes here? A membrane inside and a membrane outside. So this is inner membrane, this is outer membrane, this is inner membrane. And what is in between? Intermembranous space. Now look at the external membrane. It continues with RER. Makes sense? So perinuclear space also continues with the cisternae in RER. What is attached to RER? These ribosomes. Are you making sense? Does that make sense? Okay. Now, if you look at the inner membrane, inside the inner membrane, can you see this network of the protein, which is the nuclear lamina? Which protein is this one? Laminin protein. The laminin protein, that's the one which is forming.
The differentiation is
Sorry?
it's forming the differentiated cells.
Yeah, in the fully differentiated cell, that's where it will be sitting. So it is supporting the inner membrane. That makes sense? Now, if it is not there, the nucleus is going to lose its shape. If it's going to lose its shape, it's going to lose the functionality of it. And there are certain diseases which are related. So, in the laminin deficient, people suffer from certain diseases. One of those diseases is young children look very old. Progeria is a disease. You don't need to remember. You don't need to learn more about it. Yeah. If there is any problem in any protein, there will be consequences of it. Now, what is the nuclear pore? Look at the inner and outer membrane here. See the way they continue? See the way they continue? So they don't open this intermembranous space to this cytosolic space here. Make sense? So it's closer, but it is gated by nuclear pore complex protein. Okay. And if you look at this EM, which is a freeze fracture method of looking at it, these dots basically represent the point where the nuclear pores are. Okay. Now, when you look at the nuclear pore complex, nuclear pore complex is found by inner and outer rings. Okay. Now, they form a symmetrical uh, core, and this is the space where the things from inside go outside or from outside to inside. Look at this protein here. This is the outer ring and this is the inner ring. Okay. This is the cytoplasmic ring and this is the nucleoplasmic ring subunit. Okay. Now, inside of the nucleoplasmic complex, look at this arrangement of the protein which is forming nuclear basket. Now, this nuclear basket is the one which is basically transporting things from inside to outside. That makes sense? From inside to. Similarly, we have protein outside cytoplasm. They can help things move from outside to inside. But it is not a free flow. It is being checked. So it's selective. Not everything inside will go out. Not everything which is outside can go in. It is a selective barrier. What is nucleolus? Nucleolus is the non-membranous region within the nucleus, and it's 0.5 to 5 micrometer in diameter in mammalian cell that you can see. And this is the primary site for production of ribosomes. Why do we need the ribosome? So should the nucleolus be prominent in cell forming the protein? Yes. Should the rough endoplasmic reticulum be prominent in cell producing protein? Yes. Makes sense? That's how we can see that the cell is active or cell is not active. Okay. Now, some cell could have more than one nucleolus, like some cells could have more than one nucleus. Okay. This is the type of the condensation of the ribosome. This will stain. Ribosomes have RNA acid. If you treat it with a basic dye, they will appear darker. They appear intensely. Intensely darker. With which dye? H&E preparation we are talking about. Okay, which are hemotoxylin and eosin. Now, what is chromatin? Sorry? Genetic material. Genetic material, which is the DNA and all its associated protein, which is helping it to be organized first and also division and everything. So everything, the machinery will be the chromatin. It's called the chromatin. Now, how does the chromatin look like are distinguished in macroscopically? There are two varieties of it. One is called euchromatin, other one is called heterochromatin. Hetero. Euchromatin is a finely dispersed granular material in the electron microscope, and in light microscope it appears lightly basophilic. Lightly basophilic. Okay, let's go back.
What is that?
Bluish. Base, baso, base, basic, basic dye, having more pH, alkaline. Okay, more pH, alkaline, basic. It will attach to acid because something acid will attract base. Something base will attract.
The nucleolus involved in protein synthesis, but isn't that a job for the ribosome?
That's what they have to do. You're not. That's what the ribosomes are produced. That's why they are involved. Okay. Make sense? So, if you stand, now let's look at this area there. This is the nucleolus. These areas are also appearing basophilic. Look at this area here. Compare it with this area. Look at this area here and compare it with this area. They are both basophilic, but one is dark and the other one is lightly stained basophilic. Both have DNA material, but one is more densely and one is more faintly stained. One is darkly stained, one is lightly stained. That lightly stained area is the area where the euchromatin is. Okay. So can I borrow wire? Can you all see this wire? Yes. If I stand it and show it to you, okay. Now what I do, like I'll file it. Same thing, just being coiled will appear dark. When you see this, it will appear darker. Then the same thing, when it is expanded, it appears lighter, stained but lighter. That makes sense? Yeah. This is how euchromatin and heterochromatin. So this is how euchromatin will look like. This is how the heterochromatin will look like. So if you look at under electron microscope, these are the heterochromatin. This is the nucleolus, and these areas are where the euchromatin will be. Okay, euchromatin is active. It's like when you receive your friend. How do you receive? With open arm. So you're active. Make sense?
So is it the same thing? Chromatin and heterochromatin?
Yes. Yeah.
One of them is spoiled on the other.
Yeah. When a hetero is coiled, U is used. Okay. So
Is it that?
Sorry? A small one. Yeah. We'll come to that. We'll come to that. Let's go to apoptosis. How many of you heard this term before even? Wow. Now this is a cellular suicide, but it is programmed. It is a suicide program.
That's not very quiet.
Interestingly, think about it. When we were born, before that, our face was being structured. We had a nose. Okay? We had the same nose. The size has increased from when the time we were born. But at the time of the birth, the tip of the nose stopped at the point. Even after that, it starts getting bigger. Think about it. The tip of the nose, the cells in the tip of the nose, they don't stop growing. What will happen? We will have many tips of the nose here, few heads sticking up. Can you imagine that? Let's say like a snout we will have. But no, after a certain density, they stop. Look at the way that our hand develops. Between the fingers, there were tissue that died. So we have now if that tissue between the fingers, if it doesn't die, what will happen then? We'll have kind of will be like a duck then. Some people do have. Okay? You might feel like the toes or the fingers are kind of going. Okay? Now this is the programmed cell death, which is a physiological process, and it is the one which is important for eliminating defective or not needed cells. Okay. Defective cells are cells which are not needed, they die. Now, when they die, they end up in forming the membrane-bound apoptotic bodies, and these apoptotic bodies are being taken care of by just like the napkin. We use it, and then you put it in the bin, and somebody will clean the bin, or you yourself clean the bin. Okay. Material which is not needed, the cells will die. How do they? Can you see these cells here? These are apoptotic cells here. I want to show you the simpler diagram as well. Okay, these apoptotic cells will die into smaller apoptotic body, and cells which can phagocytose those cells, the neighboring cells are macrophages, eat those cells and store these response. So finish. Okay. Now this is controlled by a family of protein. Now that family of the protein need to be suppressed, so we can have the apoptosis. Okay, that makes sense. So it's controlled by other set of proteins. Now, if those proteins are more, then apoptosis will not take place. If the apoptosis not take place, then we have abnormal growth. Okay, that's what some of the conditions, like cancerous conditions, will end up with. Okay, let's see how long. Now, I want you to read the last line. Apoptosis, the apoptotic cell, they do not rupture. They do not release their contents. That makes sense? They do not release their content. They kind of shrink and they break into pieces. They don't swell and bust.
What happens to that cycle?
Right?
What happens to that cycle?
It has been eaten up by the cell itself, is degraded, and the bleed out. You know what the meaning of the word bleed out is? Bleed out. For instance, from a balloon, you take a pinch from the balloon and take a small piece of the balloon out. Okay, it's like a bubble gum. You blow it, and sometimes people can just separate it. Okay, so you take that part out. This is how the apoptosis will take place. What would happen in case of the necrosis, which is a pathological process? So let's look at that. Look at the cell here. This is the suicide. This is the homicide.
Okay.
How? We have to have some kind of injury, hypoxic or hypothermic injury to the cell. You see what is happening to the side of the cell?
Yeah.
The cell membrane is injured and it is swollen and it ruptures. The cell organelles go out and there will be inflammatory response. As opposed to apoptosis.
Yeah.
They're pretty small and they are being taken care of. Make sense? This is how these are those cells which are showing the signs of apoptosis or the necrosis. They start swollen. Okay. So swelling and lysis is a feature of which process? Shrinkage and debris. One is more physiological, other one is pathological. Cell cycle, you must have learned about it. We'll quickly go over some of the main steps of the cell cycle. Okay. What is the end result of the cell cycle? So production of the similar cells, two daughter cells. Okay. Now, four distinct phases our cells go through. Mitosis is a very short period of the time. Mitosis itself is a short duration. Now, there are certain cells, after the mitosis, they will go into gap. That's where they function. That's where the interface. This is the phase in which cells are functioning. If a cell has to produce a hormone, this is the time. But when the cell is dividing, at that time, everything else shuts down. It only concentrates on division. Now that's why this phase is important. Okay, the interface. Now interface. These G, the letter G stands for gaps. Okay, so we have a gap 1. Some cells will leave the cycle and will become differentiated and will never divide. Give me an example of a cell.
Cardiac cells.
Neuron.
Nerve cells. Cardiac cells.
Muscle cells. They might have some kind of regenerative power, but mostly they will increase in size. That makes sense to you? But other cells like
Hepatocytes.
are there.
Hepatocyte.
Sorry?
Hepatocyte.
Hepatocytes is a good example. They have the capacity to divide many times of need. They can divide. Gut lining. Keep dividing, okay? Keep dividing. Can you imagine in 7 days' time, entire gut is refreshed with a new uh, cells inside. Okay? They will be continuously in this cycle then. Okay? So G1 phase is the phase where the DNA is duplicated. I'm sorry, after the G1 phase, which is the gap phase, we will have S phase where the DNA will be duplicated. If the DNA starts duplicating, cell has to divide after a short while. Otherwise, this cell will become abnormal as well. You cannot have more chromatin material. Okay, you can have two nuclei, but you cannot have no nuclear, and diploid, tetraploid number of chromosomes inside. Okay, so you have to then. What happened after the S phase? There is a short gap period, and then we have mitosis. So,
I have a question. During the phase, procreate and so when the phase happens, and when the phase happens in the kingdom.
no, those are the phases of the mitosis. We'll come to that. You don't have to know the phase.
So how the DNA replication?
DNA replication has DNA replication happens in the cells which decide to divide. So I am in my office. I'm in an office working. Okay, but now I have been asked to give a lecture. So I prepare for that and deliver the lecture. So a cell will be functioning normally. Then that cell has been asked to divide. That will go into
DNA.
DNA replication, synthetic phase. Then we'll have a gap 2, and then it will enter into the mitosis. Okay. Now all these phases together are called as interphase. The mitosis itself is a very short duration of the whole cell cycle. Okay. Now, if you look at the mitosis, this is the time where you can see some of the stages of the mitosis. You can see the chromosomes aligned. You can see the mitotic spindle. Someone asked me about mitotic spindle. You can see the mitotic spindles. You can see, oh, these cells are dividing. Okay, so if you look at the abnormally growing cell, the mitotic index of that tissue will be more. If you look at the cells of the skin, there will be lots of mitotic cells that you can see. But if you look at the brain tissue, would you see the mitotic cells there? No. That makes sense? Now, if you look at this one, that's where the mitotic spindle, that's where the equator, where the chromosomes are sitting. Okay, these are the two poles. They will divide. That's where the microtubule. This is kind of in the metaphase. Okay. Well, we started 15 minutes late because there's these things which didn't work.
I'm so sorry. So sorry. I will take one more minute, then you can continue. So I just have to make an announcement, and I'm very sorry to disturb you. I'm sorry that you had to start late or something was not working. Guys, today is the last day of registration. I would like to request at least 20-25 of you to drop M1 and register for M2. This cannot continue if you sitting on the floor, looking at the back. Some of you are sitting on the floor. This cannot continue. So today is the last day for you to do it voluntarily. Drop this M1 and please register on banner for M2. By tomorrow, if you have not done it and there is still this discrepancy, we will have to randomly choose somebody. We'll remove you and put you in M. Okay, thank you. And sorry again.
That's okay. Now, if you look at the mitosis, what is happening? The parent cells divide, and the two daughter cells will have a similar set of the chromosomes. Okay. Now, different phases of the mitosis. This is the prophase. What is happening in the prophase? The nuclear envelope kind of disintegrate, kind of disappear. But DNA replication has already taken place. DNA duplication has already taken place. Which phase? S phase, synthetic phase, has already taken place. In prophase, the nuclear membrane disappears. So you see that? I don't have any outline. We have these centrosomes here, pair of centrioles. Sir, do you understand now? Centrosome is a pair of centrioles. Now, from this, microtubules will come. Metaphase is the chromosomes are aligned at the equator. Can you see the chromosomes at the equator? And can you see these microtubules there? Some microtubules are attached to the chromosome at the point of the kinetochore. So those proteins are called kinetochore microtubules. Okay. Now these chromatids are pulled towards the centrosome. These microtubules kind of pull the chromatid toward the centrosome. Okay. In the anaphase. Ana means divide. Then we have the last phase, which we call the telophase. You see what happens in the telophase? Cytoplasm, the organelles, they also divide. They also divide from the cleavage, which comes from outside and pinches in the middle. So it's like you might have seen the balloon twister, the long balloon, the way that they twist, and they can cut it and then make it two balloons out of one. This is exactly how it happens. The cell will divide, nuclear material will divide first, the organelles and everything later. Now this is how it is. Look at the central dome, the spindle pole here. Look at these microtubules, which are kinetochore microtubules, and these are interpolar. Okay, so let's push each other. Push each other. If we push each other, you see where do we go? Do we come closer or do you go away? Away. These microtubules, which are interpolar microtubules, they kind of push each other. They push each other. Where the center go then? Outward, away from it. That's what the function of interpolar. What is the function of kinetochore microtubule? They'll bring chromatid. So they kind of attach to the kinetochore where the centromere was. You pull them apart, okay? So duplicated now becomes same again. Same number in the parent cell. While in case of meiosis, simple thing to understand is the DNA material duplicates once, but the cells divide twice. So we end up with haploid. Make sense? So from diploid, we divide tetraploid, but we divide twice. In case of somatic cells, what we do? We divide once. We from diploid will become tetraploid, divide once, diploid again. Make sense? Same phases, interphase, that's where they are working. That's where the cells, germ cells, they will be sitting question. Then the prophase 1, metaphase 1, anaphase and telophase 1. This phase will have the duplication. These phases will not have duplication. They will have only division. Metaprophase 2, metaphase 2, anaphase 2, telophase 2. And will end up in having 4 haploid. Each haploid will join another haploid for fertilization. So we'll maintain the number of the amount of chromosome. So we'll maintain the generation. Imagine humans have a diploid number of germ cells. So first generation will have, second generation will have, we will not have a generation after a while. Okay, if this had the case, if instead of haploid, if you will have a diploid amount in the germ cells. So how do we maintain the generations? Okay, that's why the fertilization will end up in diploid number again. Okay. Now matrix in the cytoplasm. We talked about organelle, cytoskeleton, anything else which is present in between. Aqueous substance like thing, it's a matrix. What is present there? We talked about it. Organic and inorganic substances. Organic and inorganic substances present there, and that's where the physiological processes are taking place. This is the fancy, very colored diagram. But you know everything. We talked about everything here. Some of the diseases, you don't need to remember the disease, but you need to understand the basis of it. Now, there is an enzyme needed to remove a substance from the cell that has to be present in the lysosome. If that enzyme is mutated, the gene mutation results in a faulty enzyme. Enzyme is not working. What will happen to the substrate? The substrate will build up. Substrate will build up. And that's where we will have the storage diseases. If the lipid cannot be broken down, we will end up in having more lipid. For instance, in case of Gaucher disease, that's where the glucose cerebrosidase enzyme in the liver and spleen are deficient. So the lipid will start depositing there. So what will happen to the size of the liver? It will keep on increasing because more deposition. Tay disease is autosomal recessive neurodegenerative disorder. Okay, this is due to enzyme deficiency. Okay, this enzyme when it is deficient, it starts accumulation of the gangliosides. Gangliosides start accumulating in the cells which are wrapping around the axonal processes. This happens in Jews. Canal Jews, which are a particular group of the Jews, they only marry within the family. Okay, that's why the trait is a recessive trait. Both parents are affected. In this case, what happens? Neurodegenerative, the cells which are lining the processes are affected, and then we will have auto immunity against them. Okay, there's a neurodegenerative disease. Now, these are the features of the disease. You don't need to remember the disease. I'm not going to ask you about the disease. I just want you to learn the concept here. If the enzyme is not there, there will be buildup of the substrate. If the substrate build up, it will lead to disease processes. Okay? Look at this one again. Microtubule. I want you to go back to this diagram actually. This is a cancer cell. It is dividing, and you are a new scientist, and you have been tasked to develop a drug. We need to stop this division. You need to target one of the components of cytoskeleton. Which component is a good one here? Microtubule. If you block the assembly of a microtubule, would these chromatids will move? They can't get separated. They can get duplicated, but can't separate it. If they can't separate it, the new cell be found. That's why some of the anti-cancer drugs are anti-tubulin. They are tubulin polymerization, the polymerase inhibitors. Okay, they inhibit the polymerization. Okay, that makes sense. What did we cover so far? We looked at the cell organelle, cell function. We looked at the skeleton, function. What is the most important thing that we have been emphasizing? Structure along with function. If you don't know the function, no point of knowing the structure only. You need to know the function. The last question, and then I'll open the